I'm here to talk to you today about lab safety, okay? So I always like to start out my lab safety talk by telling students this somewhat uncomfortable story from when I was a TA many years ago teaching an anatomy lab. We were dissecting cats, and we had reached a point in the dissection where we needed to pull apart the rib cage, open it up, and as you do this, little bits of cat juice, or really formaldehyde, tended to fly out. And back in those days, at this particular lab, we weren't required to wear safety goggles. After this particular incident, we were required to wear safety goggles. Okay, so one of my students is opening up the rib cage, and a little bit of cat juice flies up and hits him in the eye. And he raises his hand and he says, "Hey, I got this cat juice in my eye. Do I need to rinse it out?" And I told him, "Yes, do it immediately." All right. And it's at this point that I hear someone at the next table over say, "Are you freaking kidding me? I got it in my eye 15 minutes ago." All right, so the point here is that if something happens, tell me immediately. If you're doing these dissections at home, though, that's not an option. What you need to do is look out for your own self-interest, okay? If you get this stuff in your eye, wash it out immediately. Proper protocol is to hold your eye underwater, running water from the tap, for 15 minutes. And that'll probably irritate your eye and it'll be uncomfortable, but you've got to do it just to wash this stuff out. It's nasty, this formaldehyde. Most of our specimens are going to be stored in this stuff called Carolina's Perfect Solution, but it's really formaldehyde, some proprietary blend containing formaldehyde. And then other specimens will be stored in just flat formaldehyde. You may remember that formaldehyde is in embalming fluid. We use it to preserve corpses at the funeral home. And it is just fine for corpses because they're dead. But it is highly carcinogenic and it is really bad for living people. As proof of this, when I was a TA, same college many years ago, my first semester dissecting cats, again, I had warts on the back of my hand. And after many dissections in my first semester, the warts disappeared, and I was kind of confused as to why. And I looked it up, and apparently formaldehyde is used in very dilute solutions to kill warts because it, mutates DNA. Now, warts are caused by a virus. Formaldehyde mutates the DNA in the virus and kills it. It can't reproduce anymore, then the warts die and fall off. That's a happy side effect for me, yeah, but it's indicative of how dangerous formaldehyde is. It's indicative of how quickly it can mutate DNA. And keep in mind, I was wearing gloves most of the time, so probably most of my formaldehyde exposure came through the gloves. All right, you want to limit your exposure to formaldehyde. Now, I don't mean to say this to scare you away from the dissection or future dissections. One exposure, 2 exposures is probably fine. Just be sure to, at all times, wear your gloves and wear your safety goggles. Mine are over on the table there, okay? One other thing I'd like to talk to you about is blade safety. This is really important, okay? I've got my trusty scalpel here. Every semester, I catch a student holding their specimen in their hand and attempting to cut down into it. That is a terrible idea, okay? Why? Because if you cut through your specimen, your hand is right underneath it. You're going to stab yourself in the hand. You're going to introduce all the nasty animal bits plus the formaldehyde directly into your bloodstream. Not good, not fun, okay? You're going to have a bad time. What you want to do is hold the specimen flat on your tray and then come down from above, holding the scalpel upside down there, come down from above, make sure you've got your fingers well out of the way, okay? Never cut with it in your hand. Always make sure your fingers are out of the way, your hand is out of the way. All right, and one more note on formaldehyde while I've got you. If you are pregnant or nursing, get a specimen stored in ethanol, that's alcohol. It's almost as good at killing bacteria as formaldehyde is. And many colleges will either have it or be able to obtain it for you, okay? It's totally safe, ethanol is, totally safe to you and the fetus, provided you don't drink it. All right, so with that, I will take a tour of the lab and we will show you some of the safety equipment here. All right, continuing on with lab safety, we should take a tour of the lab. You should be familiar with the location of the hand washing sinks. We see them over here. If you are taking a class on campus, make sure that you know where the eye washing station is and where the shower location is. Ours is out in the hall. All right. You should also be aware of the location of the first aid station. If you are doing these dissections at home, make sure that you have some bandages nearby, okay? And of course, clean your wound before bandaging it. And lastly, if you're taking a class on campus, understand where the red biohazard bin is and where the regular trash bin is. Bags can go in the regular trash bin. All of your biological specimens, the brain, the eye, the heart, anything else should go in the biohazard box. If you are doing this dissection at home, you're not going to have a red biohazard bin. Just be sure to double bag it and take it outside immediately. You wouldn't want your dogs getting into this. You know what I mean? If you are doing any experiments that involve drawing blood with a lancet, be sure you know where the sharps box is in your lab. If you are doing this at home, just dispose of it as carefully as possible.